The Fact Check podcast is hosted by three rehab professionals with decades of combined experience. But I'm not one of them. I'm just making this intro. If you are finding the world of health and musculoskeletal care confusing and contradicting, and let's be honest, at times a bit crazy, our efforts here are aimed against all that misinformation through real research and real evidence. Let's do this, people. Nothing you hear on this podcast should be considered medical advice.
Hi, everyone. Welcome to the Fact Check podcast. I'm Alexi, aka Nobullshitphysio on all platforms. I'm a physical therapist, and I'm joined today by my co-host.
Hello, guys. I'm Skeptical Physio. You may know me also as Diego Hidalgo. You can find me on Instagram as the Skeptical Physio. And I'm a physical
today we have a
therapist
very special guest, Dr. Joe Zundell, specialist in cancer. Can you give yourself a good intro? Because I guess you're probably better at describing what you're about.
Sure, yeah. So I am a cancer biologist, so I received my PhD in cancer biology from the University of the Sciences and the Wistar Institute, actually in Philadelphia, Pennsylvania. I mean, I've got a diverse palette of scientific training from microbiological sciences to marine biology, but obviously my specialization is in understanding the foundations of cancer, aka cancer biology. I don't know how much more you need to know in that. I mean, we can get into my thesis work, which is in a specific type of cancer, but generally my entire training is established on the foundations of things that, drive multiple different types of cancers. And obviously that's extremely variant.
Yeah, obviously. All right, let's get started with a bunch of questions to get through. If you had to explain the pathophysiology of cancer very succinctly, what would you say?
Like the origins of cancer?
Yeah, the origin story. Like, how does it start? Like, the mechanisms, broad strokes. Like, why do I get cancer, essentially?
Yeah,
like
just to try and keep this simple for people,
Yep.
I would say that the generalized consensus about what cancer is it begins when cells don't continue to follow the normal rules that govern cell biology and can sustain cell growth past what is considered normal.
Okay.
And obviously the ways that it can start is again, hypervariable. It's not always from a DNA checkpoint regulatory method, because there's obviously a lot of checkpoints in terms of DNA cell cycle control and mitosis, which is obviously what governs cell division. But there's a variety of angles regarding how our immune system recognizes abnormal tissue within our body and clears cells that are on the path to becoming abnormal from our bodies to potentially developing into cancers.
Okay.
That's the simplest way to put that.
Makes sense. All right, Diego, your turn.
Yeah, so a while ago, I think like 5 years ago, I read a book on epigenetics by a biologist that you may know. He's called Manuel Esteller, and the book is called I'm Not My DNA. So I would like to ask you, what's the, let's say, state of the art when it comes to the relationship between cancer and epigenetics, if you know about it?
Yeah, actually, believe it or not, my thesis work was surrounding epigenetic regulators for a specific type of ovarian cancer called ovarian clear cell. And so there's a variety of things that can happen in cancer, and epigenetic dysregulation is one of the many things that can happen. And essentially, it's assuming that cancer has already developed usually, or we're under some level of chronic stress that enables a variety of different genes to be expressed, which can then be a predisposition for the pathogenesis and development of cancers. The link there is that there's a specific subset and environmental component associated with how genes are expressed at any given moment in time. And depending on our nutrient availability, depending on oxygen availability, those things can essentially change how DNA is read. And in relation to my own research, there's direct things that interact with chromatin. Chromatin being, DNA is very tightly organized within the nuclei of our cells. And various bodies of genes are expressed and not expressed at any given moment in time. They're wrapped around these things called histones or nucleosomes. And now the way that these are unraveled from nucleosomes and histones to enable these different things to be expressed, these different bodies of genes to be expressed, is very tightly regulated by chromatin modeling complexes. So at any given moment in time, depending on how cells are adapting to these nutrient availabilities or oxygen sensing, as an example, which we often see in cancers, they will open and close these genetic sequences based on methylation statuses and acetylation statuses to control wide bodies of gene expression. And so oftentimes in cancers, when it's related directly to epigenetics, we see dysregulation in a lot of the enzymes that control methylation. So even just controlling the interactions of DNA to histones, the quality of control there in terms of the responsiveness regarding the epigenetic responses of enabling specific gene bodies to be expressed is often dysregulated. And now we still have a lot to learn in the cancer space because epigenetics is kind of a blanket term for thousands of things happening at any given moment in time. And so that's actually why it's one reason why a lot of these inhibitors that we've developed against epigenetic processes have been relatively unsuccessful, because they're hard to target towards a specific epigenetic thing because it's involving broad strokes when it comes to regulating gene expression. Does that make sense? I know that was a little bit, that was quite a bit.
Yeah, I think if you could explain what histones are and methylation for the listeners.
Sure. So histones are protein complexes that mediate the interactions between DNA, kind of like a ball of yarn inside a nucleus. So for DNA to be organized inside of a cell to where it can save space, it has to be organized in the nucleus by being wrapped around histones, these balls of proteins, like balls of yarn. And so in order to regulate the interactions between DNA, that's where methylation and acetylation comes in. So methylation is generally associated with repression of gene expression, so suppressed genes, or an off state, like an on and off switch. So the methylation, if a gene is hypermethylated, it's associated with increasing interactions to histones. But if it's acetylated, it'll decrease the interaction of DNA to histones, making the DNA more open from the chromatin or the histones, enabling gene expression machinery to get in there to interact with specific types of genes. Obviously, there's different transcription factors that will bind to any given gene depending on sequences that they recognize. These are hypervariable depending on what the transcription factors regulate and a variety of other things.
So
does that clarify things for you, or?
Yeah, I think so. I would like to ask you also another question that is related to this one, and it's like it seems that the main, correct me if I'm wrong, but it seems that the main mechanism by which cancer develops is through some cells, let's say, avoiding the apoptosis mechanisms. And I would like to ask you, why does it happen? Why some cells can escape that?
Okay, so there's a number of ways this can happen, and one thing that I often like to teach about is the hallmarks of cancer. So that is one of, I think, 16 hallmarks of cancer is the evasion of apoptosis. And so some ways that this can happen is that cells can specifically deregulate some of the machinery associated with pro-apoptotic and anti-apoptotic responses. So there's things like BCL2, which is very important in terms of regulating apoptotic responses. Oftentimes, it's anti-apoptotic. So we will try and target something like BCL2, which is often upregulated in cancers because it's enabling the cells to evade apoptosis. So that's a direct intrinsic mechanism about how cancer cells can upregulate something like BCL2 to evade apoptosis. Another way that cancer cells can evade apoptosis is through immune cell evasion. So immune cells being, doing their job, hopefully, will be able to screen for aberrant cells that aren't quite looking normal just from a cell surface perspective. And one way that cancer cells will do this, one of many ways that we've discovered, is that they will upregulate receptors that suppress immune cell responses. So either of you might have heard of something called PD-L1. So cancer cells will upregulate this protein called PD-L1, which interacts with PD-1 receptors on something like a T cell. And it suppresses the T cells from interacting and inducing cytotoxic responses, killer cell responses, against cancer cells. So those are two methods by which cancer cells can evade apoptotic mechanisms. Is that clear?
Yes, I think so. Yeah,
that makes sense.
Thank you.
Sure. just spinning off of the epigenetics, I was wondering how close we have a lot of genetic testing for cancer. As a consumer, is there any form of like epigenetic testing, or is it only for research purposes at this stage?
1 second here. Gotta put my computer on Do Not Disturb. I keep getting pinged.
Popular.
No, actually, it's
Popular
it's more
guy
like,
alert. Yeah.
No, it's more like crappy emails that I have to delete later.
Just the IRS, like, where are your taxes? Where are your taxes?
It's like Old Navy. It's like Old Navy trying to sell me some pants I don't want.
Okay.
So your question was, are there any available tests to screen for epigenetic dysregulation? Is that your question?
Genetic markers of disease.
Yeah, so there are currently some blood tests that you can buy that attempt to analyze changes in methylation status of DNA. So it's like circulating DNA that we can look at their methylation status. I think that they're useful, but currently, and I will say that alone, they're not super efficacious. I think in order to make a prognosis for cancer, you need multiple different types of tests. These epigenetic tests can be used, but they need to be used alongside other things like, specific MRI tests or even CT scans or, PET scans, those sorts of things. But on their own, they're not super useful because you can get a lot of false positives or even false negatives from a lot of these tests because you look at these global changes in methylation status or even acetylation status, it's not always going to give you an accurate picture of cancer.
Yeah, that's why when you said methylation, my eyes kind of pricked up because it's like a common claim in the biohacking sphere. I've even seen it used as an insult. It's like, oh, you're going to get methylated or whatever if you do that.
Yeah, that's absurd to me. I mean, I see this, it's the same level of, I guess, I hate to be mean in saying this, but it's kind of like intellectual laziness. It's the same people that say that, oh, you need to balance your hormones, right? It's the same thing with epigenetics. If you say that you need to regulate your methylation status, it sounds cool, but it doesn't mean anything when you actually try and understand anything about epigenetics and how it relates into diseases, and then further realizing that even when like I said, I spent a lot of my thesis using chemical modifiers to reduce some of the epigenetic responses, and more often than not, they were very unsuccessful because of just how broad and non-specific modifying methylation patterns can be.
Yeah.
So that's often why we use combinatorial approaches. We can give like a demethylase inhibitor if it's upregulated in a specific cancer, but we usually need to use something else as well.
Yeah. Yeah, I guess spinning off of this, one researcher I really respect, I think he's at Brown, Geronimo Barajo. We'll probably have him on the pod at some point, and I probably murdered his name. He says that, I think we had a conversation where he said that essentially at this stage, precision medicine is pretty much a scam because it is maybe in the future, but right now we don't have much like things we can act on when treating, like, let's say, at least as a GP, like, oh, you need this drug because of this gene. There might be some exception, but he said that far and wide it tends to be like a scam. Would you agree with that statement, or maybe nuance it a little bit?
I'm hesitant to call it a scam just because it kind of poo-poos on some of the evidence out there that suggests that it's useful. But I am definitely in agreement that right now precision medicine is generally too primitive. Obviously, these things take time in relation to the research to be studied well enough to not only understand their safety, but also understand dosing at which things will be efficacious. And also understand like toxicity profiles, pharmacological profiles, those sorts of things. And that's something that I work with regularly because I'm also in this like precision medicine space where my current job is to make targeted radiopharmaceuticals. Obviously, I can't discuss the targets on this show, but like the nuclear medicine space, without, obviously I'll get fired and probably sued. The nuclear medicine space is really primitive. We haven't really advanced things in the radiation oncology space, since the Marie Curie times for the most part. So trying to make these things targeted comes with a lot of informing the FDA what to do and where we can realistically do it. So while I have a lot of hope in this space, I'm not really going to say it's a scam right now. We just need a lot more evidence to show how efficacious it is. And we need to be more rigorous about the science that we're doing, such that when it does come time for it to get into the clinic, it will be as useful as possible with as low of toxicity profiles as possible, while considering a specific person's need.
Yeah, I'll let Diego go, but I'll probably say also I think cancer is probably a very special case, because you could argue that cancer is like a DNA mistake disease. It's like probably the dumbest way to explain it.
Yeah,
but if you compare it to like a heart attack, it's quite different. Like, the precision aspect might, like, there's a lot of screening for like which type of cancer do you have and which treatment we're going to go for.
Sure.
Which I guess we don't have for a lot of diseases, from what I understand. Anyway, I might be completely wrong on this.
No, it's not completely wrong. I think, trying to think of cancer from a DNA perspective is important, but when we make drugs to target specific processes in cancer cells, we often have to think about where the thing is inside the cell that we're targeting. And this is true regarding any disease. But when it comes to cancer, one of the really challenging things for most cancer biologists or people making drugs is targeting transcription factors, because they're very quickly made inside cells and then they go into the nucleus. But we can't often make drugs that have high enough penetrance to get to the cancer, get internalized into the cancer cells, then interact at a dose sufficient enough to actually interact with the specific transcription factor that we're going for, because there's a bunch of other things in the cell. So usually when we try and target those things, it comes with a lot of off-target interactions that are essentially like targeting needles in haystacks of needles. So it becomes really challenging. So that's why we're often just limited by the biology. And I think that this is where something truly useful can come into play, especially regarding immunotherapies, because they're already on the surface of the cells. So things that have been super helpful in the clinic would be like these anti-PD-L1 therapies. They block the PD-L1 receptor that cancer cells are upregulating, so that now they can't suppress the T cells, and this enables the T cells, at least by one signal, to recognize cancer cells without having to get into the super specifics about internalization of that particular protein. So just having something localized on the surface of the cell makes things significantly less complicated as a biologist trying to make a targetable molecule.
Makes sense.
Okay, Joe. So we have been talking about precision medicine and whether it's a scam or a field in development. And a few years ago, I read another book by biologist Leonard Hayflick, the creator of the Hayflick limit. And the book it's in Spanish, so the title could be like "How and Why Do We Age?" So in this book, again written in 1991, it talks about the role that telomeres may have on aging. And I think that a few years ago, there's a biologist in Spain called his surname is López Otín. He's very much into cancer. So when I listen to him and to the things that he has to say, I developed this question, and I would like you to answer it the best you can. So my question would be, how telomeres are affected by cancer, and what insights can we obtain about extending our lifespan due to researching cancer?
Absolutely. So just as a quick fun fact, Leonard Hayflick actually did a lot of his thesis work and a lot of his postdoctoral work in the proximity of where I got my thesis at the Wistar Institute, which you might have seen. So Leonard Hayflick discovered this thing called the Hayflick limit, which is essentially this limit to which cells can divide. And, there's obviously some seminal papers that he had published back in the day to show that, I forgot even the cell line he used, which might have actually even been due to the Wistar rat at the time. But he established that certain cells can only divide a certain amount of time because they have so much genetic material that they can reciprocate through mitosis. But in relation to cancer, what this had enabled a lot of researchers to study, and actually one of the neighboring labs next to where I did my thesis, Dr. Jesse Villanueva, studies a lot of this stuff too. And oftentimes in cancers, we see the issue associated with telomere shortening. So in cancers, if they can't regulate a lot of these DNA-related things, they'll do so in a very poor way. So, this is often confusing for people because people assume that cancer cells, they do these really weird things to continue growing in a very calculated way. They do, but the thing is, there's a lot of pitfalls associated with this and a lot of trade-offs. And it actually ends up creating a lot more issues, which is why people end up dying from cancers. So one of these issues is because of their level of dysregulation with DNA repair mechanisms, which we often see in tumors that are genetically unstable, which is a lot of different types of cancers. They will enable telomere shortening. And so if telomeres become too short, it'll enable something called mitotic catastrophe. But even then, cancer cells continue to bypass that mitotic catastrophe and keep growing. And so this creates this like snowball effect of continuous issues where the telomeres become so short. And then you start having issues of, as these cells continue dividing, you start getting things like not trisomy, but like aneuploidy. So they'll make duplications, amplifications of chromosomes. They'll do translocations. So you might have heard of in CML, chronic myelogenous leukemia, or even AML. Is it AML? No, it's CML. You might have heard of the Philadelphia chromosome, which they'll have this translocation at a chromosome, which results in this fusion protein called BCR-ABL. And that's actually one of the first technically targeted precision medicines that were produced in the cancer biology space, because it targeted something that just wasn't being made in normal healthy cells. So it made this fusion protein due to a translocation in DNA, largely due to telomeric shortening. And we were able to target this essentially new enzyme with this drug called Gleevec, which was very, very effective, and is still even very effective to this day. And we're trying to make new drugs that eventually respond to resistance to these drugs. But that's one way that telomeric shortening can enable mechanisms in cancer that kind of cause a snowball effect of making things worse. Mm-hmm. And Leonard Hayflick was kind of a pioneer in this space, and honestly led to this aging component related to cancers.
So we have been talking about the role that cells play when it comes to cancer, and now we are talking about telomeres. So I would like to ask you this question, and it's about let's say that humans have billions of cells, and we have organisms that are much bigger, like for example elephants or whales. Do you know where I'm going to? Really?
Absolutely, yeah. I know where you're going.
Why animals with many more cells do not develop cancer in such incident rates as us?
Yeah, so I mean, it's a super good question, and it's one we don't know all of the answers to. We have a lot of hypotheses, and there's even some good data to suggest that what we know is as true as possible, right? But this is something that's called Peto's paradox, which you might have been familiar. And so the phenomenon is that these animals that have way more cells than we do often have reduced rates of cancers. We see this in bivalves. Remember, I said I had an initial training in marine biology. We see this in bivalves, clams, as an example. We see this in specific species of whales. I think the pilot whale is one of them. We see this in elephants. But regarding elephants, we know that, for example, P53 is frequently mutated in human cancers, at least 50% of the time across all human cancers. But elephants, it turns out, have more than, humans. Obviously, we're diploid organisms. We only have 2 copies of a particular gene. But it turns out elephants have multiple genetic copies of P53. And this might be, I don't say this with super confidence, but it looks like it might be, one reason why elephants have reduced rates of cancer, just because of how important P53 is in terms of regulating DNA repair mechanisms. So that's just one reason. But there's a variety of others that honestly I'm not too familiar with, but that's just, relating to the elephant for people to understand that there. Cool. And of course, there's also something to be said about the differences in environments. Not to get all like woo-woo and, we see all these things with like ultra-processed foods and toxins in our environment. Humans, we do create a lot of problems for ourselves. And I think there's something to be said about the anthropogenic effects, the human-related effects of the things that we do on our own environments that can increase our own cancer rates despite having, all of these checkpoints within our cells to help regulate us. We know that things like smoking, tobacco are not healthy. We know that chronic drinking is unhealthy. We know that chronic sleep disruption is unhealthy. And yet, with any disease, especially even in things like cancers, we have issues here. So it's definitely one thing to think about the total amount of cells that an organism has in terms of its relationships to cancer risks, but it's also another thing to understand the ecosystem that an organism occupies, because nothing is ever black and white in nature. It's always a combination of how we interact with our environment and also genetic heritable things that we might have as well.
Yeah. I will say also one thing humans are good at is we're probably the only species that's able to maximize its lifespan because we don't have predators essentially. I mean, the rare serial killer or like the guy who tries to hunt the tiger. Yeah. But in general, there's a lot of animals that if you raise them in captivity with vet staff, they live like twice as long. There's some live shorter lives. I don't remember which ones, but there's a couple animals that live longer in captivity, which cats are good examples. And it could all I'm not saying it's the only effect. Obviously it's not, but there's probably a part where it's just like we have more opportunities to develop cancer because we do use that lifespan. There's more likelihood of cancer forming.
Yeah, exactly.
Yeah.
Exactly. I mean, obviously the longer we live, the greater our chances. And especially as we get older, a lot of, things wear down as you get older. Your consistent ability to regulate a variety of cell processes as you age, it goes down. And that's just the normal process of aging. And I think people need to remember that as well in relation to this discussion.
Yeah. I do want to touch on, just really quick, we still don't know why naked mole rats don't get cancer, right? Mm-hmm. We still don't know.
I mean, I don't personally know.
You didn't ask them.
If I'm being honest.
Okay.
I can Google that. I mean, I think I've looked it up before, but I don't specialize in the reduced cancer rates of naked mole rats.
Yeah.
Perhaps we can call up Kim Possible. You guys remember that show? No?
Yeah, great show.
Yeah, there you go.
Okay. Well, we're going to do one more serious question of the general questions, and then we'll probably get into the myth busting.
Okay, cool.
So why do some cancers metastasize while others just remain there?
That's a great question. And actually, there's a really good scientist, a French scientist, his name's Joan Massaguet, who has dedicated a lot of his career to studying metastasis and things like breast cancer. And, as you probably are aware, there's not just one mechanism, but we've generally understood that, metastasis is typically a feature of late stage disease. And some cancers, depending on how genetically unstable they are, might get to that state of metastasis quicker, but it is typically a late stage adaptation because the genetic and environmental framework to enable metastasis is it requires multiple different events as a cancer progresses to adopt phenotypes where a cell can actually deregulate cell surface attachment receptors and adopt characteristics that enable increased motility, and also then adopt further characteristics to then attach to another organ and change its genomic and environmental interface from a genetic level to basically seed into a different organ and grow. So that's literally what metastasis is. But going back to Joan Massaguet's work, there's this process called epithelial to mesenchymal transition. This is a process, again, and usually seen in late stage cancers that is often driven by things like TGF-beta, which is a growth factor. And it controls a variety of different processes, not even just from maintaining cell growth, but is also very important in processes like embryogenesis when humans are developing. But these things have double-edged sword processes in terms of mediating programming for metastasis. So sometimes these growth related factors will bind to their receptor and stimulate these long signal transduction cascades inside of a cell to enable this thing called epithelial to mesenchymal transition. And in doing so, they'll downregulate things like E-cadherin, these cell surface attachment things that enable cells to be attached to their basement membrane, assuming it's a solid tumor as an example. So they deregulate these things that enable them to stick and often increase these proteases, which cleave protease attachments. So again, it's multiple things. They make these proteases that cleave attachments to their basal membrane so that they can detach. And then once they detach, they need to upregulate specific proteins like vimentin is another protein associated with a mesenchymal or motile phenotype to be able to extravasate into the blood, get through tissue, squeeze its way through into the blood vessels and travel and then get to distant organs. This is why we see often common sites of metastasis. Like, for example, a common metastatic site for lung cancer is brain. A lung cancer won't metastasize to everywhere, but it does commonly metastasize to the brain. And that's because of features that are realistic and adaptable from that lung tissue that might be present in the brain. So it doesn't have to change so much by the time it gets to the brain. It can, seed in a relatively quick manner to continue growing. And again, going back to your previous initial question, metastasis is something that happens late stage, and that's because there's a variety of things that need to happen. And I just named one in relation to TGF beta, but there's a number of different growth factors that enable signaling cascades that can start metastatic programming, but these take a lot of time in terms of developing cancer cells to adapt. It's not something that happens overnight.
So, Joe, I have one additional questions before we enter into the myth-busting part. And I would like to know, let's say, the answer to something that unfortunately happens a lot when people say, oh no, I've been cancer-free for 3 years, or I'm in remission. And then it seems that, let's say, those cancers go to sleep, but then they reappear years later. Are we talking about let's say, a new episode that it has a different origin, or are we talking about the root of the problem that keeps going at it?
If I think I understand your question correctly, it's like it's how cancer cells can exist in like a inactive state that enables recurrence of a tumor after many years.
Yeah.
And so, again, there's a lot of ways that this can happen. Sometimes it's just that the tumor cells themselves stay below a rate that is detectable, such that 3 years later, they've grown to a state where now they're starting to cause symptoms. But realistically, they might have been there all along, because I think, especially in relation to solid tumors, we can give, let's say we take a solid tumor, we do a biopsy of a specific cancer type in the clinic. Let's say it's a tumor that I studied, ovarian clear cell carcinoma tumor. We get it out from the ovary, and we do some screening on it to determine a specific target to treat with. We can treat that person's cancer depending on the things that we discover with a multitude of drugs, but the thing that people need to realize is that a solid tumor is not one type of cell. It's a number of different types of cells that have adapted differently depending on where they are in the tumor, depending on oxygen availability. And this is kind of the epigenetic thing that we went back to previously. Depending on oxygen availability, depending on nutrient availability. So cells in the periphery of a solid tumor will adapt very different than those in the inner core of the tumor that don't have as much oxygen, that don't have as much glucose or glutamine or a variety of different amino acids as an example. And so we can give a drug and it'll target most of the cells that are actually targetable by the drug, but it won't kill off the cells that haven't adapted mechanisms to be sensitive to that drug. So sometimes there will be cells left over that weren't treatable in the first place, and they're also below detection. So there are some technical limitations from a clinical perspective. So while something might look like it's in remission, we might still have some cancer cells there in a patient that has developed cancer. And so that's usually one reason why we see recurrence later, and it's usually due to chemo resistance. And so oftentimes when the cancer comes back, because it was from such a small population within that tumor, it's significantly more challenging to treat because there may not be great options for the treatment of cancer, especially because that's technically a later stage. Does that make sense?
Yes. Thank you.
Yeah, of course.
All right, on to the fun part. So you're familiar with Wim Hof,
really
right?
fun to me.
This was the fun
Sorry.
part.
Okay, so no, that's not what I meant. Yeah, you're all familiar with Wim Hof, the Iceman?
Yeah.
Okay.
The
Did you know he
Iceman guy,
cures
yeah.
cancer?
Yeah.
Okay, here we go.
What is
Yeah.
your story?
Oh,
Hi,
Jesus.
Wim. My story is 2 years ago, I got diagnosed with stage 4 incurable advanced cancer. And obviously they didn't give me a lot of hope. They just sent me home to try and live the way I could and put me on palliative chemo. But I came home and I decided that I could do something extra to go along with the chemotherapy that they had. And the brother-in-law suggested that I read your book. And obviously I read your book And it inspired me and blew my mind. And they told me not to touch anything cold on chemo. So the first thing I'd done was got in an ice bath. And, yes, and cold showers every day. I was running with no t-shirt on all the way through winter to try and get the cold exposure and doing the breath work. And my first scan that I went for, the tumours had actually started to shrink. So, yes, amazing, absolutely amazing. So I was feeling really, really good, even though I was going through chemotherapy. And I found that I wasn't losing my hair because of the cold showers. I don't know whether it's the different type of chemo I was on, but yeah, I had 35 rounds of chemotherapy, but I managed to run 6 miles every day.
Wow.
Whilst I was on chemotherapy, I changed my diet to vegan, yeah, just basically lived my best life.
Yeah, I think we're gonna pause it there. But my first comment as a non-expert is just that he did all of this, 35 rounds of chemo, went vegan, was active, and then it's like, no, it's the breath work. That's my first impression. What's an expert's opinion on this?
So this is really interesting.
Good for him, by the way. Good for him, by the way. I'm not
absolutely good for him. Like, this guy was able to live a relative, and this is me just taking what he said at face value, assuming that all of it was true, that he did follow all of these things. But at face value, we don't know what type of cancer he had. So he said stage 4 advanced cancer. That doesn't mean anything to me, because you can have the staging of cancer depending on what type of cancer it is. If I have stage 4 colorectal cancer, my options for treatment and even surgical resection will be different than a stage 4 of a glioma, which is largely untreatable. So if there is some level of truth to this, and I don't know why a doctor would say not to touch anything cold. That's a very strange suggestion that I don't anticipate any oncologist would actually say. But the thing is, like, depending on certain chemotherapeutics, as an example, and from a biological level, he mentioned not losing his hair. We know that, in the research that we've done, that a lot of chemotherapeutics will target the hair follicle because the turnover rate in cells of the hair follicle are very quick. So they utilize a lot of the same mechanisms that cancer cells will also use to grow. And that's often why cancer patients will lose their hair when they're on chemotherapy. And I saw this firsthand when my mom had esophageal cancer. But one of the things that they're implementing now is cold caps. Because if you put a cold cap on, so there's some benefits to cold showers and baths and things like cold caps for people on chemotherapies that are known to induce hair loss, because we can slow down specific metabolism rates associated with cells of the hair follicles. If somebody's got a cold cap on, or they're in a cold bath, and that can reduce the likelihood of hair loss. It'll increase the likelihood of the therapy to do its thing. But that doesn't mean that, like you point out, that the breath work and the cold therapy are doing a majority of the work. It might have been helpful, and it probably was from my scientific perspective here. Yeah, those cold caps. And these things are used widely when people are giving chemotherapeutics known to do this. But it doesn't mean that those things did more than the chemotherapeutic alone. We have to consider quality of life when we treat patients. And again, I'm not an MD, I'm a PhD. But when we do, see these things in the clinic, it's multiple things alongside chemotherapies that can enable a person's quality of life. And good for this guy to be able to, keep running and doing his thing and being as healthy as possible. And, Wim Hof, as much as I don't think he deserves credit here, there's something to be said about it. I don't want to crap on the idea just because he says crazy stuff. But the issue here is that these people pose it like it's some sort of science, but they're not utilizing the scientific method to explain why these things might help the people that they intend to help. It's just, it's kind of ignorant.
Yeah, I agree. We probably won't have time to cover it, but I have like a big post coming up about people that claim that X cured cancer. And one thing, and we see that a lot in physio, where they're like, the doctor said this, that, and like that I would need surgery, and then I did this thing and that was fine. And it's just like often I'll see doctors making prediction about stuff they have no fucking. It's like an orthopedist sees the X-ray or the MRI. He's like, yeah, you're never going to be able to run again. And I have patients that have like horrible osteoarthritis and they're performing at extreme levels. There's a paper by Eliud from 2019, I believe, where they looked at high performing asymptomatic soccer player. I believe it was like Premier League players. So pretty much the hardest thing you can do on your knees is probably play soccer at the highest level on earth.
Yeah.
And 97% of them had meniscal tears. So it's like, I see these people, so it's like almost like the certainty that either patients perceive from the doctor or the certainty that doctors exude is helping misinformation spread. Because one thing you'll say is like the doctor said at 5 months, doctor said at like 6 months or whatever, and then I survived. There's this paper here. I'll share my screen. Yeah. Basically looked at how good doctors were at predicting the survival rate for cancer. So the overall accuracy of predictions for predicting 12-week, 24-week, 36 weeks, and 48-week survival was 83%, 72%, 75.5%, and 74% respectively. So essentially, unless you're like just about to die, doctors are pretty like there's a quarter chance that they're wrong. Essentially, I'm sure they're really good at knowing that you have cancer, but it's not that they're bad at their job. It's just to some extent everyone's different. Every case is different. Like, as you highlighted, like every cancer is different. So it's just unfortunate. I don't know how doctors do it nowadays, but I feel like maybe a more responsible way to kind of phrase it instead of saying like, no, you're gonna die in like 14 days and 27 hours.
Well, I don't think it's that doctors aren't being responsible in how they're saying it. I think the problem is that we're hearing the people that were given a prognosis that didn't line up to what happened for them. We're not hearing the people that, actually had the statistics that you referenced line up. Yeah. We're seeing the people saying, trying to spread this idea that doctors don't know what they're talking about. Look at me. I did all of these things and I live past prognosis. We're not saying that, you can't beat the odds. We're saying that your likelihood of beating the odds is within this range because of the data. It doesn't mean that you can't do it because we honestly, like any oncologist will hope that you beat those odds. We want to help people. We don't want to keep them sick. And this might transition into one of your other ideas. But, it's just, again, we're seeing the minority.
Yeah. Survivorship bias, essentially.
Yeah, exactly.
Yeah. And every lottery winner I know plays the lottery, so it's a good investment. Yeah.
There you go.
Yeah. Probably the other one we can pivot on, Diego, I don't know if you agree, would be the solar callus claim. We have a Paul Saladino hot feature today for that.
Okay.
Yeah, we hate him too, but he's
we are
funny.
sorry.
It's okay. I don't hate him. I think he just needs to be quiet. We'll get into the clip.
All right, let's go.
Let's talk a little about skin cancer. As I mentioned earlier, there are three big types of skin cancers with a precursor lesion called actinic keratoses. But the three big types of skin cancer are melanoma, squamous, and basal cell cancer. Some people say squamous. Correct. I say squamous. Who knows? Now, let's start with melanoma. This is the one everyone fears. Not really a sun-associated cancer when you look at the data. So I want to start with this study when we're talking about melanoma. Skin cancer risk in outdoor workers, a European multi-center case control study. If you look here at the conclusions, you'll see that though outdoor workers had more risk behavior and they had less health literacy, they had more exposure both occupational and leisure, and they had less sunscreen use, they had a higher risk of developing the sun-associated cancers, actinic keratosis, basal cell, and squamous, but they had no significantly increased risk of developing melanoma. So that is quite interesting, and it's important to note that this finding is corroborated many times in the literature. There is a lot of literature to suggest that there are perhaps multiple roads to melanoma, not all of which are related to the sun. Again, this study is called Is There More Than One Road to Melanoma? It's a rapid review. From The Lancet in 2004, paradoxically, outdoor workers have a decreased risk of melanoma.
Great, Paul can read.
Suggesting that chronic sunlight exposure can have a protective effect. So, clearly, getting burned when you were a kid is a bad thing.
Oh, yeah. Did you want me to pause this? Yeah.
No, I'm just saying, cool, Paul can read. All of those things are true.
Let's keep going. You let me know. Just raise your hand if you want me to pause.
And let's return to the paper that I teased you guys with at the beginning of the podcast, the melanoma and dietary lipids. Again, this is a paper from 1987. And it is fascinating because there is this association, both an increase in linoleic acid in the triglycerides of subcutaneous adipose tissue. There's an increase in 10 years between 1975 and 1985. People are eating more seed oils. We know that. And the percentage of polyunsaturated fatty acids found to be higher in melanoma patients relative to controls. Really fascinating finding that brings up a lot of interesting conclusions, or at least hypotheses, that I think we need to examine with regard to melanoma. Is it possible that increased linoleic acid consumption could be causing fragility to cell membranes, and that could be leading to oxidative damage in the sun, leading to DNA damage, and then more melanocytic nevi, precursor lesions, or melanoma? Or could the same thing be happening with squamous and basal? I would say yes. It's not supported by literature yet because there haven't been any studies looking at this. We need many more studies with linoleic acid.
We're going to pause it here. All right. Right to answer. Go.
What's the question?
So is there any validity to what he's saying? And, you know, I have no idea.
Yeah.
I have no horse.
Yeah, so, I mean, it just sounds like he's reading papers, giving people statistics about how there's, you know, no direct relationship between. My cat literally just jumped over a wall into the room. Sorry.
That's okay.
But he's basically saying how there's not a strong relationship between the sun and melanomas, while there is, or I guess UV exposure and melanomas, but there is between basal cell carcinomas and actinic keratinosis, and even squamous cell carcinoma. But, you know, he's also mentioning that there's dietary considerations in relation to melanoma rates or skin cancer rates. All of those things are true. It's hard to say whether or not it's directly because of seed oils. I know Paul has this message about, you know, seed oils, and I think there's some level of truth behind consuming a diet that's not nutritionally rich, that has maybe an excessive amount, which is relative, of seed oils. But if you consume a nutrient-rich diet, you're not consuming so much seed oils that it's going to cause oxidative damage or whatever the hell he wants to talk about. Now, I don't know what the point here is behind him talking about this, because he's not an oncologist, and I think it's good that he's trying to read these papers and maybe make people more conscious about skin health, but it's not like a just avoid seed oils sort of situation, or avoid the and I've heard him say this too, avoid using sunscreen situation, because they're lying to you, because sun isn't even involved or UV is not involved with melanoma. But just because I don't think that there's not a strong relationship between UV exposure and melanoma doesn't mean that I want to get basal cell carcinoma, which accounts for 90% of the other cases of skin cancer as well. Mm-hmm. So it's like, you should still protect your skin, but in the off chance that there is a small link between UV exposure and melanoma, I still don't want melanoma, because melanoma is still the most aggressive of those types. So if just protecting my skin with, you know, either clothing, SPF clothing, or even a sunscreen, and we can debate different types of sunscreens, but as long as people are protecting their skin from chronic exposure of ultraviolet radiation, while understanding that there's healthy levels of ultraviolet radiation regarding things like vitamin D, we can be healthy. But it's the problem with Paul and his audience is this acceptance of extremism, where people want to believe, if I cut out seed oils, I'll have a perfect diet. If I don't wear sunscreen, I won't get skin cancer, and I can become an optimal or primal human being and live the way that our ancestors did. But that's not true. And I think we all need to accept that there's this balance that needs to be achieved and maintained in life. And it's never these extremes that, unfortunately, people like Paul seem to operate on. And I don't know how I can convey this any better, because at the end of the day, it's like these people want cut and dry answers about what to believe. They don't want to think about, you know, these things critically to know what to do for their health. They just want to be told something. And I think that's kind of what Paul's audience offers these people, because they don't want to think for themselves, or they don't have the time to think about all the science about, you know, how to reduce skin cancer rates. But realistically, people need to develop the time to think about these things. It's our overall health, and it's never going to be this extreme thing like avoiding seed oils or avoiding sunscreen or sleeping at the optimal day, staring at the sun to optimize our circadian biology. It's none of these extreme things. And that lack of clarity can often make people more confused and pissed off at the healthcare system, because we can't give people those extreme answers because they're false.
Yeah, I mean, one of the claims I think a lot of people make for seed oils causing cancer or whatever is based on the idea that they're inflammatory. But I've reviewed all the RCTs on this. There's a post on my page. If you look at human trials that match the calories, so isocaloric RCTs, there's no evidence that they're inflammatory. In general, they tend to actually do the opposite. So there's a trend towards them being actually less inflammatory than saturated fats, which are advocated by people like this. The only evidence that shows this is in rats or in studies where essentially you're creating a calorie surplus. So there is some truth to it, but it's just that people tend to overconsume seed oils because they're in junk food a lot. But it's not the seed oil in there. It's just that it's this really tasty food you're going to overeat.
And it's not nutritionally good for you. Right. So if you overeat those things and it compromise or it consists of most of your diet, you're not going to be healthy. Yes, seed oils might play a role there, but it's not the seed oils alone.
Yeah.
So here's the thing. I also sorry to interrupt here, but I think
Diego's just going to sign off.
Oh, okay.
Diego, do your sign off.
Yeah, guys. So again, thank you so much, Joe, for participating in this podcast, because I had a lot of questions about cancer that I needed to ask you sometimes, and nothing much to say. But again, thank you so much, guys. If you want to find me, you can find me at SkepticalPhysio on Instagram. And nothing much to say, guys. Take care. Yeah. Thank you.
Bye, Diego. And happy birthday, man. We'll keep going for about 10 minutes, I'd say. Yeah.
Thank you, Joe.
Feliz cumpleaños, hermano. All right.
Muchas gracias. Adios.
Adios. Adios. So again, just overconsumption is, I think, a very common thread. But is there that claim that I had never heard is about the theory of the antioxidant debate? Is there any claim that linoleic acid is actually oxidizing, and that could be a potential pathway to cancer? I've never heard that. I mean,
yeah. So there are pathways from which linoleic acid is oxidized and can act as a fuel source for cancers that upregulate a lot of the fatty acid desaturase enzymes that utilize linoleic acid for metabolic purposes. But I mean, it's not like people have this assumption that everything we consume, like if I have cancer, if I'm diagnosed with cancer tomorrow, not everything I consume is going to go directly to the cancer cell. So things need to be broken down, catabolized, even anabolized in a particular series of multitudes of reactions to be used by a variety of different things in our bodies, all of our organs, our tissues.
Yeah.
And so I think people have this general idea that when I eat something like a seed oil that has linoleic acid in it, that linoleic acid is going to go all the way directly into that cancer cell that needs it, and it's going to be incorporated into these desaturase enzymatic reactions and directly promote cancer cell metabolism. You see the same thing again with Paul and his audience in relation to high carbohydrate diets, sugar fueling cancer, that sort of thing.
Yeah, I think that's one claim I hear a lot. Is there any truth? I looked into the evidence, that just like spoiler alert, so I'm really curious about your answer there to the idea that sugar fuels cancer. Because I'll tell you, I'm not going to call myself a layman, but I guess like in between, like my understanding is that if you don't consume sugar, your body's just going to produce it because that's what we use as fuel. We do this thing, I think it's called like gluconeogenesis.
Correct.
You don't have it, you're going to do it. That's when ketosis hits, I think, when your breath starts smelling weird, it's because your body's doing that, right?
That depends, but yeah, generally.
Okay. So yeah, I kind of don't understand how that could be true. That like eating, let's say I'll make my claim as precise as possible, is a sugar-rich diet with a caloric needs are met. Like you're not overeating. Is having like more carbs or more sugar actually like a risk factor for cancer? Do we have any evidence of that?
A diet that has high amounts of added sugars can predispose us for diseases like diabetes and obesity. And so then diabetes and obesity are risk factors for a multitude of cancers like pancreatic cancer as an example. And so to put this as simply as possible, just because something can operate as a fuel source for a cancer doesn't mean that, like I just said, doesn't mean that the things that I consume are going to go directly to that cancer cell metabolism because of things like gluconeogenesis. And it's going to vary depending on where the specific cancer develops. So if it's, like a liver cancer, for example, gluconeogenesis happens in specific zones within the liver.
Yeah.
So if I have a tumor that develops within this particular zone of the liver, where gluconeogenic rates are high, especially in a fasted state, glycogen stores will be broken down to help fuel tumor metabolism, or gluconeogenesis will be implemented to enable glucose production for tumor metabolic purposes. But it's not like that's going to directly come from glucose consumed exogenously through a person's diet. So yes, sugar can fuel cancer and accelerate processes, but it's not directly by consuming glucose that a person will worsen their cancer. And this is something that often you see people will say, oh, starve yourself of carbohydrates, which is much more than just glucose. Starve yourself of these amino acids. Don't eat these seed oils. Don't eat these fats. They fuel cancer.
Yeah.
But as you point out, your body will, when you stress it out in certain situations, like if you go into ketosis, your body has all these adaptive tools, even cancer cells, because they have the same mechanisms inside themselves as well. They'll implement these own metabolic cascades to make their own fuel sources. And it's also nucleic acids as well,
That makes sense. But yeah, I guess my other question is, you mentioned like the diabetes. I'm very familiar with that explanation. Did a lot of research. I read a lot of papers before doing this, and I think a really common theme is that microinflammation, and which is something that's caused by diabetes, and obesity, which is one of the reasons why they're risk factors for pretty much anything on earth.
Yeah.
I understand that mechanism, but I don't even know if like just having more refined sugar is, if you don't eat in a overconsume, like if you're not in a caloric excess situation chronically, does that actually even predispose you to diabetes? I mean, I'm sorry, I know that's kind of it's not your specific field of interest.
It's a little bit, no, I mean, I'm interested in it, but it's a little bit out of my field of expertise. So you're saying if somebody doesn't consume or hyperconsume,
so I just eat more sugar, but the same amount of calories as someone else, does that actually predispose me to diabetes type 2? Like, I don't even know. We can just cut this out if you don't know, because I don't know either.
So here's the thing. I'm cool with saying I don't know.
Yeah.
And I think people need to see both of us saying I don't know to a particular situation, because we're not supposed to know everything. In fact, we're supposed to say I don't know to stuff that I don't know.
Yeah. No, I agree. So, we'll keep this in. I will mention there is, I think we'll finish on these, because my mini literature review, I'm looking for, hang on, it's a paper on cancer, and hang on, let me...
Oh, good. Take your time. I really appreciate you getting the literature and finding these videos for this discussion. It's super helpful.
Oh, hang on. Yeah, okay. So there's only one paper. So the idea is, if cancer, you can just starve your cancer, we would have the cure for cancer. We just go on a keto diet. There's two papers on it. There's one where, and I didn't review that paper well, where it said that it helped with the treatment. Like it essentially potentiated the effects of chemotherapy, made it more effective.
Correct.
I don't think it's an awesome paper. I'm not an expert, so I'm not going to expound. There's one paper that has credible findings, I'll say, and it's this. I'll share my screen. Hang on. And I want your opinion on this. Okay, sure. So long-term effects of a ketogenic diet for cancer, 2023 paper by Ryuichiro Egashira. So I presume a Japanese person.
Yeah.
Interesting, as the other paper is also in Japanese people. So they showed that a total of 55 patients with advanced cancer were enrolled, and essentially they compare the survival rates to other people. That would be, they essentially did a case match, or not even a case match that would be higher quality. What they did is just compare it to the expected survival for these types of subcases, and they said, oh, they actually survived longer in these 55 people. And then the argument was like, ketogenic diet, that was the headline. The ketogenic diets, like, they cure cancer or they treat cancer. I think we can both see the limitations in such a design, right?
Right. And so this was ketogenic diet alongside chemotherapy?
Yeah. That's the only thing they changed. Yeah. So I'd say there's no control group, obviously. They just compare a rate, which is extremely global, and as we showed, can vary a lot, like 25% mistake rate on the survival. So I don't think that's the best design, but I do think it warrants replication. That's a very interesting finding, though. Also, but again, the better paper is this other paper by, I had it pulled up too. Hang on.
Is this the Rabinowitz study?
No, hang on. It's the Psy and all, or I think it's Is it C? Yeah. Hongling Kai. I'll share my screen. I'm probably butchering that name. Yeah. Okay. So this paper was actually awesome. So, Cancer Science paper, 2022, low carbohydrate diet and risk of cancer incidence, the Japan Public Health Center based prospective study by Hongling Kai. And essentially what they did is they looked at people who had low carb diets and they compare it. It was super huge. There were 15,203 cancer cases. And they looked at people before they got cancer. They looked at essentially their rates of cancer and they compared to their diet. It showed that low carb diets and more animal fat diets were both linked with higher cancer rates. So it was about 8% for low carb diets. You had an 8% hazards ratio. So essentially like an 8% added risk of getting cancer. And then for animal fats, it was comparable. Depending on the cancer, though, interestingly enough, there was like an inverse correlation for, and maybe you can explain that because that kind of boggles my mind. Gastric cancer, where there was a 19% reduction in the rate of that. That's the only cancer that was showed.
19% reduced risk for gastric cancer
for low carb diets.
Low carb diet. Yeah. So I mean, this is proof of concept that every cancer will benefit from something differently. And there's multitudes of studies, even outside of this one. Earlier, before you had brought this paper up, I mentioned there's this lab at Princeton, Josh Rabinowitz. He's been doing a lot of stuff in this space regarding ketogenic diets. You can look up Joshua Rabinowitz, Dr. Josh Rabinowitz.
Yeah, I'm looking that up.
And he studies and has even in the process of conducting clinical trials examining ketogenic diets combined with chemotherapeutics for pancreatic cancers.
There it is.
So alongside this study, yeah, exactly. So I think this is even an ongoing clinical trial that's still been going for a few years. But they see similar things across different cancer types. There are some diets, like ketogenic diets, which will help reduce the progression of things like pancreatic cancers or even gastric cancers or even some colorectal cancers, whereas they might make other cancers worse. So everything has like a metabolic trade-off, right? And so if I have a patient that I know has a gastric cancer or a specific type of pancreatic cancer, and it's in line with some of the observations that we see in these studies, then it might be important to consider that, a ketogenic diet might be helpful for this person. But the problem here isn't that people think about the diet alongside chemotherapy. It's people think that the diet alone will be helpful. And in many cases, at least from clinical data, and you can see based on this study, if you look at some of the figures from this study, it looks like the ketogenic diet makes some of their conditions worse. They don't show all of that data, but if you continue to look at the trends in one of their figures where one of their control groups is just the ketogenic diet in one of their preclinical models, it looks like it's making the tumors grow faster. And to me, conceptually, that makes sense. But the diets, and I've spoken about this on a number of podcasts, is the diets essentially create a different metabolic framework in cancer cells where they adapt to the nutrients that are within the new diet. And now we can sensitize those cancer cells to a therapy that relies on those cells adapting to the ketogenic diet. The normal cells will continue to do their thing with the ketogenic diet being present, but the cancer cells will change things in a different way that makes them more sensitive to now the addition of the chemotherapeutic, which can target the cancer cells more effectively. So these diets can enable a window of opportunity, but alone, from what we're seeing clinically, they're either going to make cancers worse or, ineffective on their own. So that's kind of all I have to say about that. But, there's a lot of scientists in this space, whether it's like Dr. Walter Longo, Dr. Thomas Seyfried, they come online and they confidently state these things related to diets and cancer. But realistically, a diet alone is not going to positively affect anyone's cancer. And usually it's with the intent to combine it with something that can, the cancer cell will change its metabolism to be more adaptive to that diet and then sensitive to the addition of a new type of therapy. But a lot of this is newer work. And so there's still a lot that needs to be learned here. And so there's conflicting data on which cancers are sensitive, which cancers are not. But that's the purpose of continuous acquisition of data. Like we're continually acquiring clinical observations and analyses of these data to determine where these diets can be useful.
Yeah. And for those at home that can't see the paper, I do want to specify that this was done in mice. So yeah, early stages. And it did mention an interesting finding. It said that although long-term growth suppression, well, it said that tumor growth suppression was only observed in mice that had an intact immune system. So it's not a lot of cancers. It's like the opposite. What's the cancer of the immune system? I forgot what it's called. Is it?
There's many. So it's either myelomas or leukemias.
There you go. Yeah. So yeah, and I want to go back to just to finish the other paper. I hang on, this is not the right paper. Hang on, where was it?
I will say, in relation to the last one, there are human data points now in clinical trials. Okay. But that's an ongoing thing. So we can't make strong conclusions from them because all we have right now is preclinical data. Once we get the clinical data and it's assessed by professionals, we can make better conclusions about this stuff. And of course, it depends on the amount of patients and those sorts of things.
Okay. Yeah, and just a reminder to people, the ketogenic diet overall increased the rates of cancer by 8%. So it was just this one gastric cancer. So there's a lot of cancers. It could also be, and again, this is me being extremely skeptical. It could just be a spurious result because if you have enough cancers, one's going to pop up. But there were 15,000 cases, so I feel like probably it's enough to actually have a powered sample. And I want to remind that animal-based diets were, linked with higher cases of cancer. This could be just because they tend to be overall, despite what people are saying, a little bit inflammatory. That would be my guess. Do you have any other specific guesses? And before you answer, I want to specify in the paper they show that this can be mediated by having high levels of plant fats in your diet.
Okay.
Yeah.
So, I mean, a lot of that too will also come with fiber, and we know how helpful the consumption of fiber is for reducing risks for gastric cancers, colorectal cancers, pancreatic cancer. Yeah, most human cancers can be benefited by consuming some level of soluble, even non-fermentable fiber sources. But when it comes to animal protein-based sources and cancer risks, I think a lot of that comes from, I guess, nutrient-poor diets that are high in animal-based proteins that have high levels of saturated fats that don't necessarily account for the importance of vegetables and fruits in a person's diet. So it's this lack of a balanced diet where people are consuming the majority of animal-based foods relative to, the importance of vegetables and fruits in their diet.
Yeah.
So it's not that animal products are bad. It's that they're consuming too much of them relative to other things that they should also be eating in their diet. That comes with risks.
Yeah. That makes sense.
Yeah.
I'd say maybe it's different for, it's so complex. We all have to remind people we're talking about cancer. It might be different for heart disease. We had Adrian Chavez on, and it seems like it's over consuming animal fats, even if you compensate with other things like vegetables. It's not like you're just improving your risk some places, but you're not actually reducing how bad it is. It'd be like smoking and taking like omega-3 and being like, it cancels out. It doesn't really work that way.
It's definitely not true. And it's always the same with cancers too. It's like there's always going to be these positive and negative benefits behind, the behaviors that we choose with our diets and anything in our lifestyles that's going to affect things and not necessarily translate to risk reduction with other diseases. So yeah,
that's a good one. Can I just ask like one more question, maybe like a broader question?
Dude.
Yeah. So I think we should conclude by, obviously I'm a physical therapist. For MSK rehab professionals, is there anything that you'd want us to know about cancer, how to treat it? Like, how would I treat someone who has cancer versus someone who doesn't, besides the obvious human condition aspect, showing empathy a little bit more, maybe?
Can I ask you a little bit of a question here?
Sure.
So have you worked with cancer patients?
I worked in oncology. I had a rotation, for like a couple months.
Okay. So how did you guys go about treating cancer patients, if you don't mind me asking?
We would just exercise, and then as much as possible without the patient puking. And or if they were crying, like we'd be like, okay, we'll come back later. Right. Okay. Pretty common. Like you see a guy my age, stage 4 cancer, he maybe doesn't want to. Yeah, so you'd have to. Mood was very, very important for these patients. Yeah.
So I mean, of course, from your perspective, paying attention to a person's behaviors, being empathetic to their condition, but also I think from a PT perspective, we need to better understand. And by we, I mean I don't necessarily understand all of the things that you would have to go through in your own training here, but I think we need to better understand the physical limitations of cancer patients. And I was actually just on Pam Greshik's, I don't know if you follow her, but Pam Greshik's podcast, and we were just talking about this regarding PT. And, one cancer patient's mobility will definitely be very different from another patient's depending on surgeries they've had, treatments, if they're currently on treatment. And so I think when it comes to cancer patients, PTs need to be very adaptive to what that cancer patient is going through at that moment in time, and shifting their exercise plan to enable them to continue to get exercise while not injuring themselves. And that could honestly be said about anybody. But with cancer patients, obviously, the likelihood for injury is a lot higher because of all the things that they're currently going through. That's the only thing on top of being empathetic that I could realistically say. It's obviously an area of outside of my expertise, but I think nowadays we're starting to incorporate physical training into cancer patient recovery now more so than ever. And I think it's a really important message that it needs to be individualized, just as you'd have an individualized training and nutrition plan for, an athlete. The same considerations need to be met for a cancer patient or anyone with any particular disease.
Yeah, that makes sense. Like one thing I learned recently of a patient who has had cancer for 30 years essentially. Chemo and other cancer drugs, they're pretty bad for your musculoskeletal system. It's essentially like a cortisone injection, but you only get the side effects. So skeletal wise, obviously it's great. It treats the cancer. Fantastic. Right. But it doesn't provide pain relief, which can be a bummer because you're treating someone, they're doing well, and then it's like, I want you to survive. So obviously you get the chemo, but their shoulder's doing worse. And that's it's definitely true regarding conditions like, and I've spoken about this on many podcasts about combating cachexia, which, as you point out, is often caused directly by the thing that we're treating with. So of course we don't want the chemotherapeutics to cause muscle wasting, which is what cachexia is. But actually, believe it or not, more often than not, cancer patients will often die more from cachexia than they will the cancer. Obviously, there's a multitude of things in relation to the drugs and the cancer itself, but muscle wasting creates such a frail state that at some point you can't even give a person a treatment because they're so frail. So if you combine a person's treatment regimen to stabilize and maintain muscle mass throughout their treatment, not only will their prognosis be better, but their response to treatment will be better. And we're actually seeing that from a mechanistic perspective regarding how exercise affects immunogenic outputs in response to immunotherapies, but also even just from an understanding of musculoskeletal physiology in progressing cancers regarding myokines and those sorts of things. Yeah, there's this bit with Louis C.K. and I think it's Ricky Gervais, where Ricky Gervais is a doctor. He's like, what do you have? Is well, all of it. And it's just like, what can I do? It's like, nah, you're just too unhealthy. You can't get healthy anymore. It's going to kill you if you try. It's just you're just stuck with it. But it's interesting. You talk about cachexia. Same thing for heart attacks. I believe if you're overweight, you have a better chance of surviving because you're not going to die of essentially wasting away. But you have more chance of getting the heart attack. So it's kind of like a paradox. Medicine is weird.
Honestly, it's not though, because even going back to my own personal experiences with cancer, and I don't know if you know this about me, but my mom passed away when I was 17 from esophageal cancer.
Oh, no idea.
And that's what stimulated me to become a cancer biologist. Yeah, it sucks, but it leads me to do what I do. But the thing is, the thing that she passed away from was a heart attack, and it's because her body was so frail that she just couldn't handle anything anymore. And so it's tough to talk about, but you see oftentimes in these cancer patients, my mom was a hefty woman. She had a lot of weight to her before her disease. And I think by the time she passed, she was literally 90 pounds. My mom was 5'5", 5'6", and she was 90 pounds. And it's that's a stick figure. Yeah. And it's a combination of the medication. It's a combination of, having a late stage cancer that continuously develops and metastasizes to the bone. Continuous lack of exercise because you can't exercise because you're in so much pain. It's all of these things that eventually the heart gives up before, the quote unquote cancer takes you. But it's the cancer is the aspect that enabled this cascade of crap essentially.
Yeah. Must be tough. How does it make you feel when you see like misinformation on cancer being spread on, because it's rampant on social media, and I can't imagine your algorithm because you work in the field. Like, how does it make you feel as a person?
Yeah, the easy answer is absolutely horrible. And I started my page, I was very angry, and I think rightfully so, because you see all these people just spouting misinformation and seemingly without any appropriate foundation or understanding of biology. And I'll be the first to tell you I don't always get it right either. And there might even be things on the podcast I've said today that aren't 100% accurate, although I've done my best with my training to try to be. And as nuanced as possible, I'm telling you I don't know.
Yeah.
But seeing these things online is absolutely infuriating, and so much so that when I started my Instagram to try and educate people, I'd often do these call-out videos, and I quickly realized that doing these things leads my audience to just follow me to make these, kind of volatile videos to call out people like Paul Saladino, that they end up not paying attention to the things that I want them to pay attention to, from like the understanding the foundations of biology, but more so looking forward to me taking another shit on Paul Saladino or Dave Asprey. And believe me, these people absolutely deserve it. Mm-hmm. But at some point you have to realize, is what I'm conveying to people actually productive? Are these people sticking around for productive reasons? And I realized that a lot of people weren't. So I've shifted my messaging a little bit, or to try and be truly educational. And it's severely impacted my traction on Instagram. But in doing so, I get way better questions and interactions from the people that are clearly paying attention than before. It's like, oh yeah, Paul Saladino sucks. We already know that, right? It's not going to change. He has no incentive to change. A lot of these people have no incentive to change. But the only incentive we should have when people should come to my page is to actually learn something and expect that we're not always going to get it right. And so while it infuriates me in the background, I often just have to take a deep breath before I make content and just spit the facts to the best of my ability. And I have some videos even on my camera right now where I sit down and I do my best. And sometimes I just sit down and I have B-rolls of me just screaming at the camera for 20 minutes because I can't be calm about it. And I don't show anybody that footage, but it's for me to see, So I'm not going to act like it doesn't frustrate me. It pisses me off to no end. You can probably hear that in the tone of my voice.
Yeah.
But it's not productive for people to see that all the time.
Yeah.
I show people when I can that it does upset me, but I don't let it affect the realism that we all just need to take a deep breath and actually learn this stuff. So that way we can know when to avoid people spouting misinformation that have no intent of changing or accepting when they're wrong.
Yeah. I agree. And I think that for those that don't know, this is how this podcast came about. I was going to just shit on someone, and then you kind of told me it might be more productive to expound on the nuance on this topic. So I'm glad this happened. But also, like, this analogy I came up with recently is that you can't just push someone off of the boat into the water. You need another boat. It's like, oh, this boat you're on is leaking. It's a bad boat. And it's like, okay, well, what am I going to do? Like, swim to the other side of the ocean? No, it's like you need another boat. So that's why I still do, like, we'll call them like call-out videos. Less and less. I try to attack ideas more, but sometimes it's just such a good example that, like, recently someone said she's not going to cite a study because a lot of studies are biased. So I just showed, well, yeah, if you cite the study, we can actually show who funded the study. Like, this is the only way we can know is if you cite it. So I think, teaching little lessons and explaining what the person said is true, because what she's saying is true. Funding does impact studies a lot more than
it does.
Yeah, I haven't cited that paper because I find the results kind of worrisome, but no one's covered it yet. But I probably will.
Send me the paper? Just send me the paper. I'll take a look at it too.
Yeah, it's crazy. The number, I don't recall the exact number, but I think it's significantly more likely to be for a study to show positive results if it has industry funding. I think a lot of people will interpret that as Big Pharma is telling you what to put it to cook the books. Right. It's more likely that they just give their money to papers that are well done that are more likely to show result.
Yeah, I mean, the thing is, so I'll talk about this from my own perspective of being in academia. So when we get our PhDs in cancer biology, I've written grants. Yeah. Right. I got my own F-31 funded when I was a thesis graduate student. I scored in the 7th percentile to get my grant funded by the National Cancer Institute. When you write a grant, you present your information in a way to say, I found these observations in this specific cancer type. I want to pursue these avenues to better understand it and from a clinical perspective, provide some sort of level of translation about how I can target it to treat a cancer. Do you think that the National Cancer Institute is going to fund your grant if you sit there and you say, I found this really interesting thing that actually doesn't work, and I can't really target in cancer, but I'm not going to be able to treat it. But I think that this negative outcome is super important. Do you think that their likelihood of funding that is more likely to fund the work that I show an avenue of targetability versus something that's not going to work?
Yeah.
It doesn't mean that they won't fund the negative data. It just means that we need resolution to problems. And oftentimes, if we fixate on the things that don't work, it can be a distraction. So it's the same. It's independent of being funded by pharmaceutical companies. Because at the end of the day, we need to get things into the clinic that have a high degree of likelihood of working to treat the thing that we're trying to do. It doesn't mean that we should ignore negative data. I would absolutely love negative data to be published. It would have helped reduce all of the things that I had repeated in a lot of my own experiments that could have just been published. That would have saved me a lot of time. But the truth of the matter is we need positive outcomes to be able to push things forward more than we do negative outcomes. It doesn't mean that they can't inform positive outcomes, though.
Yeah. Yeah. And I'm not going to pretend like there's no dark shit happening. There's probably some nonsense happening.
Of course.
But yeah, I agree. It's more the way you described it. So the analogy I would use is that it's like seeing A24 produce films win a lot of Oscars. Your conclusion could be, there's corruption. They're funding the Oscars or whatever. They're sending them to trips to Bora Bora. Or you could just think, hey, like the movies that A24 funds are only good movies because they know they're good at picking movies. You could say the same about the pharma industry. They're not like two kids in a trench coat, because there's a lot of research. Like I reviewed a paper today, it got published in a journal, and they said the effect was significant, but it overlapped with zero. That's like stats 101. Pharma is not going to fund this. They have way too much money at stake. So again, not saying they have clean hands, but it's probably more to do that they're good at funding papers that are going to have good results. And selecting.
I will say at the end of the day, it depends on the scientist, because like I've worked with amazing scientists and I've worked with not so amazing scientists. I've even been in positions where I've severely messed things up and I've been not so amazing of a scientist.
Yeah. But
we make those mistakes, right? And we try and be better every day. But, expecting that this issue persists just because of, funding is a very short-sighted one. And while it can be a predictor of some outcomes, it's a big picture, but it's not the only picture.
Yeah. I'll mention, you talk about messing up. I have this funny anecdote. It was not so funny at the time. As a master's student, I was working in the lab. We were basically looking at the effects of exercise, like an acute bout of physical exercise, on memory. Like basically learning a motor task, how good you were performing if you did exercise, like very intense exercise right after, like a 50-minute intense bike ride. So we had to measure the VO2 max of participants. And I had done it in a while because we had a lull in the summer. And I had to go back, and I didn't really remember how to do it. I pull in the cable. It's just this tiny little stupid cable that does like the inflow that calculates the VO2. I pull it, and I'm like, oh shit, the machine's not working anymore. I text my boss. I'm like, what do I do? He's like, this cable was worth $15,000. Like, $15,000, there's no replacement. This like VO2 machine is worth $150K. And I was like, what can I do? He's like, you go home or I'm fucking firing you, right? Or something. He's like, I'm failing you or something. But yeah, I passed on everything. But I've never been that stressed in my life.
It's interesting with those machines. Like, I work with really expensive machinery on a regular basis. And it's those little things that are super expensive that they don't make in bulk that, are the difference between something working and not working.
It's crazy.
And it's so easy to mess those things up, or it's so easy, like if it's a tubing, right? It's so easy to get clogged or those sorts of things if it's, something like flow cytometry. But some things are obviously more expensive than others. Yeah. But it's crazy.
Yeah. I'll just finish with the paper I was talking about. So this paper that I mentioned was the Bandari 2004 paper, Association Between Industry Funding and Statistically Significant Pro-Industry Findings in Medical and Surgical Randomized Trials. So I'm going to give the numbers. People are going to freak out, but I will say I have a friend who works in pharma. Apparently it changed drastically in the last 15 years. So it's much better now, like less of this stuff happens. But apparently if you look at the effect size, 1.9 to 8 times more likely to get a published result for it to be positive if it is has industry funding. So, it's just remain critical, like maybe favor papers that do not have industry funding, but it is to some degree unavoidable if you want to look at the totality of the data. Yeah, sorry.
Sure. Sorry to interrupt you. Here's the thing we also need to consider about industry work too. I've been in academia for a long time. Yeah. Obviously, I got my PhD in academia, and the level of funding to do academic work is logarithmically different, lower than the amount of money we're given to do work in biotech settings. So right now, I'm at a small startup biotech, and it's still way more money. Like, our company just entered our Series B, and we got $175 million to do stuff. When you get the most competitive of R01 grants from the National Cancer Institute, they're like, yeah, here's $500,000. Yeah. We have $175 million. So my likelihood of doing good work, assuming all of us are good scientists, and I will say that we are all very good, the likelihood of doing good and truly rigorous science, despite any sort of error that happens naturally with experimentation, is going to be so much greater than what you can accomplish in academic settings. So that's also one reason why you might see higher funding being associated with more positive outcomes and greater likelihood of those papers being published, just simply because we can do more rigorous science because we have more money to do so.
Yeah, I agree. Yeah, that makes total sense. That's why it's good that we're in a podcast so we can have a nuance just because if this were real, people would just be like, fuck science. 1.9 to 8 times, yes. Never follow industry funded studies. So I think it's good that you're adding the nuance. And speaking of podcasts, we've greatly exceeded your allotted time. So Dr. Joe, thank you for coming. Is there anything that you want to plug, and where can people find you?
I mean, most people will find me through whatever you distribute here, but people can find me on my Instagram at Dr. Joe Zundell. I have my own website, drjoezundell.com. I don't sell anything. I don't push anything. I have a newsletter people can sign up for through my Instagram. There's a link tree there. You can access it later for people in show notes or whatever. But also people can sign up through my website if they scroll to the bottom of the page. My goal is literally just to educate people and try and be respectful in how we receive education from each other, knowing that, at the end of the day, there's things that I'm going to get wrong. There's things that everyone's going to get wrong. But as long as we show up and have the respectful discussions and often hard discussions, I think that's how we make true impactful change. So that's what I seek to do on social media. And in doing so, implementing better behaviors about how to reduce our risks where possible, understanding that no matter if I have the perfect lifestyle, I'm still potentially going to get cancer at some point.
Yeah.
And I would have done my best. And even when I do get cancer, with the knowledge that I provide people, that there are effective and amazing treatments that can continue to prolong my life and enable quality of life if I should get a disease like cancer.
Beautifully put. Thanks, everyone. I'm the Nobullshitphysio on all platforms. This was the Fact Check Podcast. Give us a 5-star review on whatever platform you're listening to if you want. Otherwise, do not rate us at all. If you want to give us anything else than a 5, get out of here. All right. Have a good one.
10 stars.
10 stars if you can. Yeah. All right. Have a good one. All right. Bye-bye. Thank you, Joe.
See you guys.